Welcome back to another episode of Facilitate TV. I'm joined by Renee Hart, President and CBO of Lumicite. Thank you for being here.
Thank you for asking me. This is wonderful. I've always enjoyed watching your video clips on LinkedIn. You're like the news guru, you biotech guru, everything guru. It's just wonderful to watch your career grow, Ben.
Oh, well, thank you very much. Excellent. So we were talking before just with regards to like where does the industry go from here? We've gotten to some promise, but ultimately at this point we've now got to figure out how in the world do we actually reliably get these to patients at scale. That kind of seems to be one of the themes here at the show. What are your thoughts with regards to that?
Yeah, no, absolutely. It is single-handedly the next mountain the industry has to climb is how do we get to commercial scale? And there's a lot of factors that kind of feed into that. One of them is better qualification of the starting material, both the vectors, the cells used to manufacture the vectors, and the starting cells, whether it be from healthy donors for allogeneic or patient material from autologous cell therapy patients. And understanding, if possible, in a predictive way, which of those cells are going to produce the highest quality manufacture of a cell therapy downstream. Yeah. And if we looked at the vectors just in the beginning, qualifying the starting banks that are used to manufacture those viral vectors is incredibly important. You can have high viability, at the 90+ level, and that's your metric. So you think, go team go, we're ready to go, let's seed and start manufacturing that vector. But in reality, you get low yields, you don't understand what changed. Last week we got high yields, we got the same viability metric, and so there's a lot of variability in the cells that current analytics such as viability just is not telling. And so we at Lumicite are doing a lot of work in that area. It's something that we're, I guess, uniquely positioned because we are a label-free assay. So we're measuring the intrinsic biochemical and biophysical properties of these single cells, and that fingerprint, that phenotypic fingerprint, we see apoptotic trending. Those early apoptotic proteins being produced, those early membrane blubbing in the cells that other assays don't see, we see it in our assay. So that allows our customers to go, hold up, we don't want to see these cells. Let's start over and go back to our bank.
Right. Well, and I imagine, that's also a really rich data set that continues to allow you to learn more about your process and continue to optimize in a really positive direction.
100%. Gotcha. 100%.
Very good. So another key component that we discussed as well, is the quality of vector. I remember when I first started in 2020 in the gene therapy industry, people really only cared about VG. A couple of years later, the full empty ratio is getting a lot more attention, and now at this point it's really like even full empty isn't enough. You got to have a really rigorous characterization of what your vector quality is looking like, and obviously optimizing
the titer and the potency of that vector.
Yeah, optimizing for those full potent vectors. So I'd love to understand from you guys what are some of the trends you're seeing in that space.
Yeah, no, it's interesting. We cut our teeth in the vaccine market. Our early days at Lumicite were viral infectivity, viral quantification. And just last year, ATCC used Radiance, our laser force cytology instrument. They used it in their certificate of analysis for the Ad5 reference material, viral vector. That was huge for us. We were like, wow, just to see the precision of our tool being recognized in a certificate of analysis. Yeah. That's a really big milestone. And ATCC is continuing to advance. They want to be at the forefront of innovation and advanced analytics, and so they're continuing to drive even further. But that was, a big milestone for us. So now, back to the viral vector, that's one example of viral vector where we're contributing, but also we're selling into the pharma companies who are manufacturing the lentivirus, themselves. So that cell bank qualification and understanding, like we talked earlier, is a really big part of that, because we are able to tell them which of those cells are going to deliver the highest product yield. So now that we've monitored the transfection, with 5-minute answers all that time, now it's time to look at the potency of that viral titer. Yeah. So that being able to have a very precise viral titer assay is super key, because plaque and TCID50 and old antiquated assays for functional infectivity are just not cutting it.
Right. So yeah, with that being the case, right, if we're not only looking at starting material, we're looking at vector quality, but then in the context of cell therapy, right, if I have an autologous batch that's coming in and I'm going to be looking at my cells as they're going through that process, what are some of the things that you guys are seeing that might be interesting to know about whether cells are going to be a good batch or a bad batch?
No, it's such a great question. I love the autologous value proposition because to me this is game changing for patients. So we have a large pharma that is trying to get us into one of their phase 3 clinical trials. But to do that, you have to be at the hospital collection site, right? And that has its own complexities and then the clinical trial complexity. So it may be this year, maybe next year, but that is a goal. And so it's great to see the industry seeing this value. But here's why this matters. So the apheresis collection is very difficult for patients, very difficult. It's 6 hours, it's, and they're already worn out. So if you could assess their cell quality with a small sample, 5-minute answer, yes, the patient meets the tolerability bounds, proceed with apheresis, you're setting that patient up for success. Right. The pharma company knows that clinical trial slot is used effectively, and it actually is helping their clinical trial with a higher outcome of results.
Wow.
Right? So now let's go back and say, okay, that patient maybe didn't meet that tolerability bounds, for a healthy, or not healthy because they're sick patients, but for a tolerability bounds for the phenotypic profile. That doesn't mean no. That means come back tomorrow. Right. So then you ask the doctor, not us, but the physician and their team has to game plan and go, okay, do we need IV fluids, IV vitamin C, IV vitamin D, and B12 for 24 hours to boost those cells a little bit more, so then they are a little bit stronger to achieve a strong manufacture at the end. To me, that is setting patients up for success, and it's an application near and dear to my heart.
Wow. Wow. Well, I guess on top of that, right, like that even
that's kind of adaptive manufacturing, right?
Well,
like that's the holy grail.
Exactly, but that translates as well to like batch failure rates and the overall cost of these therapies too, right?
Yes.
Yeah, because you know exactly, you have a much better understanding this batch is going to work. There's no surprise halfway through being like, well, we've dropped 40 grand in reagents on this thing and that's gone.
100%. The same application holds true for allogeneic as well, because the sponsors are coming to us going, Renee, you need to get Lumicite into the collection centers because we're getting these, we have to buy 25 apheresis packs and we only need only 3 to 5 work. Right. So they buy 25, but only 3 or 5 work. So that means they have to library all those. They have to maintain them. It's just extra burden and cost associated, right? Pharma's telling us they will pay the same amount of money for the 20 as the 25, but only send them 3 or 5, right? Just qualify them better. And so that is something that we're doing now too. We're working with some of the blood centers and we're really getting out there, but how, getting the prediction qualifier in the C of A is very powerful. So looking at that phenotype of a healthy donor, because, our cells, are healthy, we think, right? Yeah. But maybe 0 of us can manufacture a healthy allogeneic CAR.
Right.
But why? We don't know. But there are some intrinsic cellular properties that current, antibody-based assays don't capture, so to speak. So that's an interesting area that we're working in as well.
Very good. And then I guess for looking into the future now, what would you say has got you most excited with regards to efficiently scaling these therapies to millions of patients?
All of the above that we just kind of talked about. But I think too, what gets me really excited is seeing the regulators supporting the industry. You're seeing a big shift where the guidance, which has been out for a few years now, maybe 2, for CMC for advanced therapies and the potency guidance for advanced therapies, they are asking the industry to feel confident to do more. Don't be afraid to change, right? Yeah. Like the history of relying on precedent, of, well, they use this biomarker or they use this assay, so I'm going to use it and you should approve it. They're kind of scrapping that because if we're going to hit commercial scale, we have to better understand our process and our product. And the only way to do that is through comprehensive analytics. And so in those guidance, they recommend matrix approaches. And we talked about that earlier today too, Ben, where, if you choose a large assay of suite of assays, KSQ Therapeutics is my favorite. They have a 12 assay matrix approach. To me, that's an insurance policy, right? Because some of them that they thought were characterization assays, over time, they were the most consistent assays, and they flipped those to potency assays. And then the potency assays flipped to characterization assays. The FDA gives you a hug either way. You are doing your job, right? You are trying to understand your process and your product. And so, to me, that is exciting, because I think the industry has always been afraid of the FDA and afraid of regulators, but they just want to have a conversation. And we were really lucky. We were invited to attend JP Morgan this year, and we got to sit in the front tables with Marty McCary giving the keynote at JP Morgan a few weeks ago. And he was talking about, we want you to come in and talk to us before you anchor to processes that may not be the right ones. And then you have to go back and spend a lot of money to redo them. Let's meet in advance, and try to let us help you answer questions. They're not going to tell you how to do it because it's your product. They want you to choose your best assay or, characterization, quantification approaches. But they can at least answer questions and help guide you as best they can. It's really a partnership at the end of the day. They want to keep the product safe and effective. And so do the developers.
Yeah, it's been interesting to see the FDA almost become, to shift and roll a little bit, instead of being like when you're 16 years old, you get your learner's permit to drive, and they're starting to act a bit more like a driving instructor as opposed to the driving tester. Obviously, they still have that role.
Well, great analogy, Ben.
And so that is encouraging for me to see, I would say.
Yeah.
Okay, so we're in a nutshell. Tell me how the Lumicite device works. Like, it's label-free, allows you to look at vectors, cells. Like, what's the underlying technology here?
Yeah, so laser force cytology is the underlying technology of Lumicite, and our instrument is called Radiance. We're a fully 21 CFR Part 11, manufacturing QC instrument. It can be used in process development, AD, manufacturing, and QC. Starting material characterization, which we kind of walked through earlier. But the most beautiful thing is it's 10 minutes of sample prep because it's label free. So you don't have all that laborious, labeling, to do, so to speak. And then, so 10-minute sample prep, 5-minute TTR, time to result, so that's a in-process, real-time optimization of understanding where your system is in the current, manufacturer. And then at the end of the day, when it gets to QC and even in manufacturing, what you care about is the user experience. So you just have to be a smart high school graduate to run Radiance, which is incredibly valuable to the workforce, right? We're really trying to bring more and more young students into biotherapeutics and biomanufacturing, but not all of them, are college educated. And so what can we do to simplify the user experience, but also keep the quality and the precision of the output? So we're cell agnostic. If you're measuring viral infectivity for a vaccine, for in-process, titer measurements, for fill and finish, for safer vaccines, right? We're not over-formulating. That's a huge application. CAR T killing assay, a potency read. We do that, and do it very, very well. There's just a lot of different applications. Whatever you're doing to a cell that you need to measure its response, stem cell differentiation, doing a lot of work with stem cell developers right now and manufacturers where it's moving into QC. We're going to be in submissions, for stem cell manufacturing, and that's a big deal.
Fantastic.
So very excited.
Excellent. Well, Renee, always a pleasure.